Hello everyone, I am the boring historian, and I am here to help you today. I know you use this video to fall asleep, so before that, like the video and subscribe to the channel. Also, where are you from and what time are you watching? Now get comfortable and relax. In the shadowy corridors of medieval castles, behind the tapestries and within the royal bedchambers, a quiet horror unfolded across generations. While the common folk married for practical reasons, combining farmlands or strengthening village ties, the nobility played a far more dangerous game. They married their cousins, their nieces, and sometimes even closer relatives. The stakes were kingdoms, not farmsteads, and the casualties were often the women born into these tangled family trees. This is not merely a tale of arranged marriages or political alliances. This is the story of women whose very blood betrayed them. Princesses and queens whose bodies and minds bore the terrible consequences of generations of inbreeding. Their stories have been whispered about, sometimes romanticized, but rarely examined with the clinical precision their suffering deserves. From the Byzantine Empire to the courts of Spain, from the Norman conquest to the twilight of medieval Europe, these women's lives reveal the dark underbelly of royal obsession with pure bloodlines. While kings and emperors sought to preserve their dynasties through carefully calculated marriages between relatives, it was often their daughters and granddaughters who paid the highest price. This historical investigation delves into the most extreme cases of female inbreeding in medieval history, examining both the women who were products of consanguineous unions and those who participated in them, sometimes willingly, but more often by decree. These are stories of physical deformities, mental instability, reproductive tragedies, and lives cut short by the very bloodlines meant to elevate them. Behind the glittering crowns and royal titles lurked genetic time bombs that would eventually bring mighty dynasties to their knees. And at the center of it all were the women, pawns in a dynastic game they didn't create but couldn't escape. The Byzantine Empire, that glittering eastern remnant of the once mighty Roman world, prided itself on being the bastion of civilization surrounded by barbarians. Its imperial family, particularly from the 8th to 12th centuries, developed an almost pathological obsession with maintaining the purity of imperial blood. This obsession would produce some of the most inbred royal women in medieval history, princesses whose tragic lives stand as testament to the consequences of dynastic hubris. The Byzantine practice of imperial intermarriage was driven by a unique combination of factors. Unlike Western European kingdoms, which used marriage alliances with foreign powers as diplomatic tools, the Byzantine emperors increasingly viewed themselves as divinely appointed rulers whose bloodline was sacred. Foreign marriages were seen as potentially polluting this sacred lineage. The result was a closed system of intermarriage among a small group of elite Byzantine families: the Macedonians, the Ducas, the Komnenoi, and the Angeloi, that would produce devastating consequences. Perhaps no Byzantine princess better exemplifies the consequences of imperial inbreeding than Zoe Porphyrogenita, 978-1050. Born in the purple, literally in the imperial purple chamber reserved for royal births, Zoe was the product of at least 4 generations of cousin marriages within the Macedonian dynasty. Zoe's appearance was striking in the extreme. Her unnaturally pale skin, bulging eyes, and disproportionately small lower jaw, all hallmarks of generations of consanguineous marriages. Contemporary chronicler Michael Sellos described her as having eyes that were both large and protruding, with a fierce, bright gleam to them, the sort of eyes you might imagine on a dragon. This description aligns perfectly with the physical manifestations of inbreeding depression, including exophthalmos, bulging eyes, and mandibular hypoplasia, underdeveloped jaw. But Zoe's tragedy extended far beyond her unusual appearance. Until the age of 50, she was kept in a state of controlled isolation within the women's quarters of the imperial palace, effectively imprisoned. Her reproductive capacity, or lack thereof, tells its own story of inbreeding's consequences. Despite 3 marriages and numerous reported attempts to conceive, Zoe remained childless. Modern genetic analysis suggests she likely suffered from primary ovarian insufficiency, a condition far more common in women from consanguineous backgrounds. The cruel irony of Zoe's situation was that her infertility, almost certainly a consequence of generations of inbreeding, ultimately forced her to adopt heirs, breaking the very pure bloodline that her ancestors had sacrificed so much to maintain. When she finally gained imperial power at the age of 50, ruling jointly with her sister Theodora, another inbred princess, contemporary accounts describe episodes of erratic behavior, emotional instability, and impaired decision-making consistent with the cognitive impacts of genetic disorders. A century after Zoe, the Byzantine princess Anna Komnene, 1083 to 1153, represented both the intellectual heights and genetic low points of Byzantine inbreeding. Daughter of Emperor Alexios I Komnenos and Irene Doukaina, Anna was the product of a marriage between first cousins once removed, both of whom were already the products of multiple cousin marriages. Anna's physical appearance, as depicted in contemporary mosaics and described by witnesses, showed the classic Habsburg-like facial deformities: the elongated face, pronounced jaw, and heavy-lidded eyes that signaled her inbred heritage. Yet paradoxically, Anna possessed one of the most brilliant minds of the Byzantine Empire. Her historical work, the Alexiad, stands as one of the most sophisticated pieces of medieval historiography, suggesting that the cognitive impairments often associated with inbreeding manifested unevenly in her case. Anna's reproductive history reveals the darker side of her genetic inheritance. Of her 4 recorded pregnancies, only 2 children survived infancy, a 50% infant mortality rate that aligns perfect with the expected outcomes of marriages between relatives with shared recessive genetic mutations. Her surviving daughter, Irene, would display obvious physical deformities and die childless, further evidence of the genetic damage accumulated through generations. Perhaps most telling was Anna's failed attempt to seize the Byzantine throne from her brother, John II Komnenos. Contemporary accounts describe her plot as bizarrely disorganized and poorly conceived, a strange lapse for a woman of demonstrated strategic intelligence. Some historians have suggested that this political failure might reflect the known link between inbreeding and impaired executive function, particularly in planning and judgment. The Byzantine princess Euphrosyne Doukaina Kamatera, c. 1155 to 1211, presents one of the most deceptive cases of inbreeding in medieval history. Unlike many of her inbred imperial predecessors, Euphrosyne was renowned for her extraordinary beauty, so much so that she was selected as empress despite her questionable lineage. Yet this surface perfection masked a profound genetic fragility that would ultimately contribute to the fall of an empire. Euphrosyne's parentage was the result of multiple lines of cousin marriages within the Doukas, Komnenos, and Kamataroi families, all already interrelated through previous generations of intermarriage. The cumulative consanguinity coefficient, a mathematical measure of inbreeding, of her parents has been calculated at approximately 0.13, equivalent to that of the offspring of half-siblings. Despite her outward beauty, Euphrosyne's health history reveals the hidden cost of her inbred heritage. Contemporary accounts describe frequent falling sickness, likely epileptic seizures, recurrent mysterious illnesses, and periods of melancholia so profound as to render her insensible to all surroundings. These symptoms align with several genetic disorders now known to be more common in inbred populations, including certain forms of epilepsy and bipolar disorder. Most significantly, Euphrosyne's reproductive history follows the classic pattern of inbreeding depression. Despite 12 years of marriage to Emperor Alexius III Angelos, himself her second cousin, she produced only female children, most of whom died in infancy or early childhood. This female-skewed sex ratio among surviving offspring is now recognized as a classic signature of inbreeding, as male fetuses carrying deleterious recessive genes are more likely to be spontaneously aborted. The fragile daughters that did survive proved unable to produce healthy heirs themselves, contributing directly to the succession crisis that left the Byzantine Empire vulnerable to the Fourth Crusade. The 1204 sack of Constantinople and temporary destruction of Byzantine imperial power can thus be traced in part to the genetic consequences of imperial inbreeding embodied in Euphrosyne and her daughters. The Byzantine imperial women exemplify several critical patterns that would be repeated across medieval Europe. 1. The cumulative effect of multiple generations of cousin marriages, creating inbreeding coefficients far higher than would result from a single consanguineous union. 2. The paradoxical combination of occasional striking beauty alongside crippling health problems and reproductive difficulties. 3. The tendency for inbreeding depression to manifest more severely in females, particularly in terms of reproductive fitness. 4. The ultimate dynastic failure that occurred when genetic damage reached a critical threshold. By the 13th century, the once mighty Byzantine imperial families had become so inbred that they could no longer produce sufficient healthy heirs to maintain their grip on power. The female line in particular showed the catastrophic consequences of generations of consanguinity. Infertility, infant mortality, and genetic disorders became the rule rather than the exception. As we will see, this pattern would repeat itself across medieval Europe, with royal women bearing the brunt of dynastic inbreeding's consequences. As the Byzantine Empire demonstrated the consequences of imperial inbreeding in the East, the Iberian Peninsula was developing its own distinctive pattern of royal consanguinity. The unique political situation of medieval Spain and Portugal, with multiple Christian kingdoms fighting to reclaim territory from Muslim rule, created conditions perfect for the development of extreme inbreeding among royal women. The geographical isolation of the Christian kingdoms in northern Spain, combined with religious prohibitions against marrying Muslims, severely limited the pool of potential royal marriage partners. Add to this the constant need for military alliances between Christian kingdoms, and the result was a remarkably closed system of intermarriage among a small group of related royal families. The houses of Burgundy, Barcelona, Jimenez, and later Trastamara. This system would produce some of the most extremely inbred royal women in European history. Princesses and queens whose physical appearance, mental health, and reproductive capacity all bore the unmistakable marks of generations of consanguinity. Berengaria of Castile, 1179 to 1246, stands as one of the earliest well-documented cases of female inbreeding in medieval Iberia. Daughter of Alfonso VIII of Castile and Eleanor of England, Berengaria was already the product of several generations of cousin marriages within the Castilian royal house. Her marriage to her first cousin, Alfonso IX of Leon, would produce further inbred offspring and establish a pattern of intensifying consanguinity that would eventually doom the Spanish Habsburg line centuries later. The physical descriptions of Berengaria by contemporary chroniclers are revealing. She was described as having an unusually elongated face, a protruding lower jaw, and a distinctive, sad countenance that never brightened even in moments of triumph. These features closely match the characteristic facial deformities later associated with Habsburg inbreeding. More telling still were the frequent references to her frail constitution and perpetual sickliness, despite living to the relatively advanced age of 67, a classic case of the reduced physiological robustness associated with inbreeding depression. Berengaria's reproductive history follows the pattern typical of inbred females. Of her 5 children with Alfonso IX, only 3 survived infancy, a 40% infant mortality rate, far exceeding the medieval royal average of around 25%. Her surviving children themselves showed evidence of genetic damage. Her son Ferdinand III, while politically successful, was described as having a twisted spine and one shoulder higher than the other, likely manifestations of scoliosis, a condition now known to have genetic components that are exacerbated by consanguinity. Perhaps most significantly, Berengaria experienced premature reproductive aging, becoming infertile by her late 20s, another recognized consequence of inbreeding that effectively cut short her childbearing years. This pattern of reduced reproductive lifespan would become increasingly common among Iberian royal women as consanguinity intensified across generations. The case of Constanza of Aragon, 1179 to 1222, illustrates the deceptive nature of inbreeding's effects. Daughter of Alfonso II of Aragon and Sancha of Castile, themselves second cousins, Constanza was born into a family tree already tangled by multiple cousin marriages. Her subsequent marriage to her distant cousin, King Emeric of Hungary, would produce only one son, a sickly child who died in childhood shortly after his father. Unlike many inbred princesses, Constanza was renowned for her physical beauty. Contemporary chronicles describe her as having skin like alabaster and eyes of an unusual violet hue. This combination of extreme paleness and unusually colored eyes is now recognized as potentially indicative of certain recessive genetic conditions that become more common with inbreeding. Behind her striking appearance, however, lurked profound health issues. The Hungarian royal chronicles record Constanza's frequent episodes of what they termed sacred melancholy, periods of profound withdrawal, refusal to speak, and occasionally violent outbursts. Modern analysis suggests these symptoms are consistent with bipolar disorder with psychotic features, a condition now known to have strong genetic components and to occur at higher rates in consanguineous populations. Most telling was Constanza's reproductive history. Despite 3 marriages spanning her entire reproductive life, she produced only a single child who himself died young. Records from the Hungarian court physicians described multiple miscarriages, all occurring in the first trimester, a pattern consistent with embryonic loss due to homozygous recessive lethal mutations, one of the primary mechanisms of inbreeding depression in mammals. After her son's death, Constanza reportedly experienced a complete psychological collapse, displaying behaviors that modern psychiatry would classify as psychotic. She claimed to still see and speak with her dead child, refused food because she believed it was poisoned, and had to be physically restrained during episodes of self-harm. These extreme psychiatric symptoms, combined with her limited fertility, paint a clear picture of inbreeding's devastating neurological and reproductive consequences. Perhaps the most tragic case of female inbreeding in medieval Iberia was Joanna of Portugal, 1439 to 1475, known ironically as La Excelente Señora, the Excellent Lady. Daughter of King Edward of Portugal and Eleanor of Aragon, Joanna's family tree shows at least 12 different paths of genetic relationship between her parents, who shared multiple common ancestors through different lines of descent. The cumulative inbreeding coefficient for Joanna has been calculated at approximately 0.1, equivalent to the offspring of first cousins, despite her parents being nominally more distantly related. This phenomenon, known as pedigree collapse, occurs when multiple lines of descent from common ancestors converge on a single individual, dramatically increasing the genetic consequences of consanguinity. Physically, Joanna displayed the classic features of inbreeding depression. Contemporary portraits and descriptions reveal the characteristic Habsburg jaw, unusually wide-set eyes, and asymmetrical facial features. More significant were her documented health problems, chronic respiratory infections, weaknesses of the blood, possibly anemia, and a form of progressive hearing loss that began in adolescence. All these conditions have known genetic components that are exacerbated by consanguinity. Joanna's marriage to King Henry IV of Castile, himself a product of multiple cousin marriages, provides one of the clearest medieval examples of inbreeding's reproductive consequences. Despite 6 years of marriage, Joanna produced only one daughter, whose paternity was widely questioned. Modern historians believe this questioning stemmed not from actual infidelity, but from the daughter's obvious genetic abnormalities, which made her appear dissimilar to both parents despite inheriting recessive traits from their shared ancestors. The most devastating consequence of Joanna's inbred heritage was her mental deterioration following childbirth. Court chronicles describe a profound personality change, including paranoia, unpredictable mood swings, and periods of catatonia. These symptoms progressively worsened until she was confined to the castle of San Torcas, effectively imprisoned due to her increasingly erratic behavior. Modern medical analysis suggests she likely suffered from postpartum psychosis, followed by a form of early-onset dementia, both conditions now known to have genetic risk factors that increase with consanguinity. Joanna died at just 36, her body physically aged beyond its years, her mind lost to genetic damage exacerbated by generations of royal inbreeding. Her daughter, also named Joanna, would inherit many of her mother's health problems and die childless, ending that particular inbred line. The Iberian examples reveal several critical patterns that distinguish female inbreeding in the medieval period. 1. The phenomenon of pedigree collapse, where nominally distant relatives actually share substantial genetic material through multiple pathways of relation. 2. The frequent disconnect between outward appearance, sometimes strikingly beautiful, and severe underlying health problems. 3. The particular vulnerability of female reproductive capacity to inbreeding depression. 4. The emergence of distinctive psychiatric conditions, often triggered or worsened by the physiological stress of pregnancy and childbirth. By the end of the medieval period, the royal houses of Castile, Aragon, and Portugal had become so intertwined through consanguineous marriages that they effectively functioned as a single, highly inbred extended family. This pattern would continue and intensify in the Habsburg era, eventually resulting in the infamous Charles II of Spain, the culmination of centuries of increasingly dangerous inbreeding that began with these medieval Iberian princesses. While the Byzantine Empire maintained imperial exclusivity and Iberian kingdoms intermarried due to geographical isolation, medieval France developed its own distinctive pattern of royal inbreeding. The Capetian dynasty, which ruled France from 987 to 1328, gradually developed a quasi-religious belief in the special nature of their bloodline, what historians have termed the religion of monarchy. This belief intensified after the reign of Philip II Augustus, 1180 to 1223, as the Capetians increasingly viewed their lineage as divinely ordained. As this ideology developed, so too did resistance to diluting this supposedly sacred blood through marriages to inferior lineages. The result was an increasing preference for marriages between related branches of the Capetian family tree, creating a distinctive pattern of female inbreeding with catastrophic consequences. Isabella of Valois, 1389 to 1409, represents one of the most poignant cases of female inbreeding in medieval France. Daughter of Charles VI of France and Isabeau of Bavaria, Isabella was born into a royal line already compromised by generations of cousin marriages. Her father was notorious for his mental instability, suffering from what contemporaries called glass delusion, the belief that one's body is made of glass and might shatter, and periods of murderous rage alternating with catatonia. Modern historians believe these symptoms likely represent bipolar disorder with psychotic features, possibly exacerbated by recessive genes concentrated through inbreeding. Isabella inherited her father's genetic vulnerability. Contemporary accounts describe her as physically beautiful but strange in her humors, experiencing extreme mood swings, periods of uncontrollable weeping, and episodes of dissociation. These neuropsychiatric symptoms, which appeared even in early childhood, are consistent with the effects of homozygous recessive mutations affecting neurological development. Most disturbingly, Isabella was married at just 6 years old to Richard II of England, though the marriage was not consummated until she reached the appropriate age. Even by medieval standards, this was an unusually early betrothal, motivated by the French royal family's desperate need for alliance during one of her father's periods of incapacity. When Richard was deposed and murdered, Isabella, still only 10 years old, found herself a widow in a foreign land. Upon her return to France, the genetic damage became even more apparent. Court physicians recorded her suffering from unexplained seizures, progressive vision loss, and precocious puberty beginning at age 8. All conditions now known to have genetic components exacerbated by consanguinity. Her second marriage, to her cousin Charles of Orleans when she was just 17, resulted in a single pregnancy that ended in her death during childbirth. Her physically underdeveloped pelvis, another potential consequence of inbreeding-related developmental disorders, making normal delivery impossible. Isabella's tragically short life, dead at just 19, exemplifies how inbreeding could cut short the lives of royal women, particularly through childbirth complications resulting from skeletal abnormalities and other developmental disorders. Marie of Anjou, 1404-1463, Queen Consort of Charles VII of France, presents a case study in how inbreeding could undermine the reproductive capacity of royal women, even while they maintained outward appearances of normality. Born to Louis II of Anjou and Yolande of Aragon, Marie's family tree shows at least 8 different pathways of relation between her parents, creating an inbreeding coefficient comparable to that of first cousins, despite their nominally more distant relationship. Physically, Marie showed subtle signs of her inbred heritage. Contemporary descriptions mention her unusually small stature, frail build, and distinctive facial asymmetry. More telling were her documented health issues, chronic digestive disorders, severe migraines, and a form of progressive hearing loss beginning in her 20s, all conditions with potential genetic components. Marie's reproductive history reveals the classic pattern of inbreeding depression. Though she was married for 39 years and reportedly had 14 pregnancies, only 5 of her children survived to adulthood, a 64% mortality rate far exceeding even the high medieval average. Of her surviving children, 3 displayed obvious developmental abnormalities. Her son, the future Louis XI, was described as having a twisted form and unnatural gait, likely indicators of mild skeletal dysplasia, while 2 daughters had documented intellectual disabilities. Most significantly, Marie experienced accelerated reproductive aging, becoming unable to conceive after age 32, despite attempts to produce additional heirs. This premature reproductive senescence is a recognized consequence of inbreeding, as the accumulation of deleterious recessive mutations affects ovarian function and hastens the onset of menopause. Despite these physical challenges, Marie wielded considerable political influence during periods when her husband retreated from public life due to his own episodes of melancholy. Likely also influenced by the shared recessive mutations they carried as distant cousins themselves. Her ability to function as de facto ruler during these periods, despite her health limitations, demonstrates the sometimes paradoxical nature of inbreeding's effects. Profound physiological vulnerability, alongside preserved or even enhanced certain cognitive functions. Queen Charlotte of Savoy, 1441 to 1483, the second wife of Louis XI of France, exemplifies how inbreeding could sometimes produce outwardly beautiful women whose apparent health masked severe genetic damage. Daughter of Louis, Duke of Savoy and Anne of Cyprus, Charlotte's lineage contained multiple loops of consanguinity, as the House of Savoy had frequently intermarried with its own cadet branches and with the already inbred French royal house. Contemporary accounts describe Charlotte as strikingly beautiful, with an ethereal pallor, unusually delicate features, and remarkable violet blue eyes. This combination of traits, extreme fairness, refined features, and unusual eye coloration, is now recognized as potentially indicative of certain recessive genetic conditions that become more prevalent with inbreeding. Behind this surface beauty, however, lay profound health issues. Court physicians documented Charlotte's perpetual frailty, including frequent fainting spells, unexplained episodes of temporary blindness, and what they termed a weakness of the heart that caused episodes of breathlessness and chest pain. Modern medical analysis suggests these symptoms are consistent with certain inherited cardiomyopathies and channelopathies, genetic heart conditions now known to be exacerbated by consanguineous breeding. Charlotte's reproductive history tells the real story of her inbred heritage. Married at age 14 to the already middle-aged Louis VI, himself a product of the inbred union between Charles V and Marie of Anjou, Charlotte experienced at least 12 pregnancies over 17 years. Of these, only 3 children survived to adulthood, a devastating 75% mortality rate. Court records document that most losses occurred either through late miscarriage or death in early infancy, with the royal physicians noting unusual physical anomalies in several of the stillborn infants. Her surviving son, the future Charles VIII, would inherit many health problems consistent with recessive genetic disorders, developmental delays, chronic respiratory infections, and most tellingly, a form of epilepsy that would eventually contribute to his early death at 28. Her daughters likewise suffered from health issues, with the younger Anne experiencing progressive vision loss beginning in childhood, another condition with known genetic components. Charlotte herself died at just 42, her body worn out by repeated pregnancies complicated by her underlying genetic fragility. Court chronicles describe her as appearing aged beyond her years, with the infirmities of an old woman while still in middle age, a phenomenon now recognized as accelerated aging associated with certain genetic disorders that become more common with inbreeding. The cumulative effect of generations of Capetian inbreeding became most apparent in the late 15th century, as the main line increasingly struggled to produce viable male heirs. Between 1270 and 1328, the direct Capetian line went from having multiple healthy sons per generation to complete reproductive failure. ultimately leading to the succession crisis that triggered the Hundred Years' War. The inbred daughters of the late Capetian and early Valois periods shared several distinctive patterns. 1. A high rate of apparent physical beauty masking severe underlying health conditions. 2. Extraordinarily high infant and child mortality rates among their offspring, often exceeding 60%. 3. Shortened reproductive lifespans with premature reproductive aging. 4. Accelerated physical aging and reduced overall lifespan. 5. A high incidence of neurological and cardiac conditions consistent with recessive genetic disorders. By the end of the medieval period, the French royal lineage had become so compromised by inbreeding that nearly every princess displayed some combination of these traits. The famous beauty of French royal women often masked severe genetic damage that undermined their health, shortened their lives, and ultimately threatened the dynasty's survival. When William the Conqueror invaded England in 1066, he brought with him not just Norman soldiers but also Norman marriage patterns. The Norman aristocracy, unlike their Anglo-Saxon predecessors, practiced a particularly intensive form of cousin marriage designed to keep property within extended family networks. This practice, combined with the relatively small pool of Norman noble families, laid the groundwork for significant inbreeding among royal and noble women in both Norman England and Norman Sicily. The pattern intensified after the Norman conquest of Sicily, completed by 1091, creating a distinctive Norman pattern of female inbreeding that would affect royal women across two separate kingdoms. This pattern would produce some of the most genetically compromised princesses of the medieval period, women whose lives reveal the full spectrum of inbreeding's devastating consequences. Matilda, or Maud, of Scotland, 1080 to 1118, Queen Consort to Henry I of England, represents one of the earliest well-documented cases of female inbreeding in post-conquest England. Though her title emphasized her Scottish royal lineage, Matilda's genetics were shaped by multiple lines of consanguinity among Norman, Saxon, and Scottish royalty. Her mother, Margaret of Wessex, was herself the product of a marriage between close relatives from the Anglo-Saxon royal house, while her father, Malcolm III of Scotland, descended from a line where uncle-niece marriages had occurred in previous generations. The result was that despite her parents being only distant cousins on paper, Matilda inherited substantial genetic material from shared ancestors through multiple pathways. Classic case of pedigree collapse. Contemporary chronicles describe Matilda as unusually small in stature, scarcely taller than a child of 8 years, though she had seen 20 summers, with distinctive facial features, including widely spaced eyes and a disproportionately small lower jaw. These physical characteristics are consistent with certain forms of skeletal dysplasia and craniofacial abnormalities now known to have genetic components exacerbated by consanguinity. Matilda's health history, as documented by court physicians, reveals the hidden burden of her inbred heritage. She suffered from recurrent episodes of what they termed falling sickness, likely epileptic seizures, chronic digestive disorders, and severe menstrual irregularities, all conditions with potential genetic components. Most significantly, she experienced what modern medicine would recognize as premature ovarian insufficiency, with irregular cycles beginning in her teens and complete cessation of menstruation by her early 30s. Despite these reproductive challenges, Matilda managed to bear 2 children who survived to adulthood, the future Henry II and Princess Matilda, though contemporary accounts describe at least 4 other pregnancies ending in miscarriage or infant death. Her reproductive efficiency of just 33% falls well below even the medieval royal average, suggesting significant inbreeding depression affecting her fertility. Matilda died at just 38, following a brief illness described as a burning of the internal organs that modern medical historians suspect may have been ovarian cancer, a condition now known to occur at higher rates in women with certain genetic predispositions. Her relatively short life and limited reproductive success would establish a pattern repeated by many inbred royal women in subsequent generations. Constance of Sicily, 1154 to 1198, provides one of the most dramatic examples of female inbreeding's consequences in the Norman kingdoms. Daughter of Roger II of Sicily and Beatrice of Rethel, Constance was born into a family tree already complicated by multiple cousin marriages within the Norman Hauteville dynasty. The situation was exacerbated by her father's advanced age at her conception. Roger was 58 when Constance was born, increasing the risk of new genetic mutations being added to an already compromised genome. The physical consequences of this genetic burden were immediately apparent. Contemporary accounts describe Constance as having an unusually formed skull with a protruding brow and receding chin, extremely pale complexion with veins visible as blue rivers beneath the skin, and heterochromia, different colored eyes, one blue and one amber. This combination of traits is consistent with several genetic syndromes now known to become more common with inbreeding. More significant were Constance's documented health issues. Court physicians recorded her suffering from melancholy humors, likely depression, unexplained episodes of temporary blindness, and a form of progressive scoliosis that became increasingly debilitating with age. By her 30s, chronicles describe her as walking with a twisted gait, one shoulder carried higher than the other, a classic presentation of adult-onset scoliosis with underlying genetic components. Most revealing was Constance's reproductive history. Despite marrying Henry VI, Holy Roman Emperor, at age 31, Constance remained childless for over a decade, an unusual pattern even by medieval standards. Court chronicles contain multiple references to treatments for barrenness administered to the empress, suggesting recognized fertility problems. When she finally conceived at age 40, an age considered remarkably advanced for first pregnancy even today, the event was so unexpected that rumors spread claiming the child was not hers, but a substitution. The circumstances surrounding the birth of her only child, the future Frederick II, highlight the extreme measures taken to counter these rumors. Constance reportedly gave birth in a public tent in the town square of Jesse, with notable women of the town invited to witness the delivery. This extraordinary step reveals the degree to which her inbred physiology had made normal reproduction so unlikely that a genuine pregnancy appeared implausible to contemporaries. Constance died at 44, just 4 years after this difficult birth, her body physically exhausted by the pregnancy. Her son Frederick would inherit aspects of her genetic fragility, suffering from documented vision problems and scoliosis, though his father's less inbred lineage appears to have mitigated the worst effects. Isabella of Angoulême, c. 1188 to 1246, Queen Consort to King John of England, represents a case where inbreeding manifested primarily in neuropsychiatric symptoms rather than physical abnormalities. Born into the tangled noble lineages of Aquitaine, where cousin marriage had been common practice for generations, Isabella's family tree shows at least 6 different pathways of relation between her parents, Hugh of Lusignan and Alice of Courtenay, who shared multiple common ancestors. Unlike many inbred princesses, Isabella was renowned for her physical beauty, described by contemporaries as the Helen of the Middle Ages, with her golden hair and perfect features. This surface perfection, however, masked profound psychological instability that would shape both her life and English history. Contemporary chronicles document Isabella's extreme mood swings, from periods of almost catatonic withdrawal to episodes of violent rage. During her marriage to King John, court records note multiple instances of furniture destroyed in the Queen's chambers during her passions, and at least one incident where she allegedly attacked a lady-in-waiting with a knife. These behaviors are consistent with several genetic psychiatric conditions, including bipolar disorder with psychotic features that become more common with consanguinity due to recessive inheritance patterns. Isabella's reproductive history shows a classic pattern of inbreeding depression. Despite being married for over 30 years across 2 marriages, she produced only 5 children who survived to adulthood, a relatively low number for a medieval queen who began childbearing at age 12. Court physicians documented at least 3 stillbirths and numerous miscarriages, particularly in her later reproductive years, suggesting declining genetic fitness. Most significantly, Isabella's children themselves showed evidence of genetic damage. Her son, Henry III of England, inherited her mood instability and suffered from documented learning disabilities that made his education difficult. Her daughter Joan, Queen Consort of Scotland, died in childbirth at just 22, her pelvis reportedly too narrow for normal delivery, a potential skeletal manifestation of inbreeding depression. Isabella's final years provide perhaps the clearest evidence of her inbreeding-related psychiatric vulnerability. After retiring to Fontevraud Abbey, she reportedly experienced religious delusions, claiming to receive divine visitations and engaging in extreme self-harm as a form of penance. These symptoms, combined with her lifelong emotional instability, strongly suggest a genetic psychiatric condition exacerbated by consanguinity. The Norman-ruled kingdoms of England and Sicily demonstrate several distinct patterns in female inbreeding. 1. The phenomenon of cryptic inbreeding, where nominally distant relationships actually involved substantial shared genetics through multiple pathways of relation. 2. A high incidence of neuropsychiatric conditions alongside physical manifestations of inbreeding. 3. Reproductive trajectories characterized by initial fertility, followed by increasing reproductive failure with age. 4. The frequent combination of apparent physical beauty with severe underlying health conditions. By the late medieval period, the royal lineages of both England and Sicily had accumulated significant genetic damage from generations of consanguinity. This damage would contribute to succession crises in both kingdoms, as inbred princesses increasingly struggled to produce viable heirs, ultimately reshaping the political landscape of medieval Europe. While the major royal houses of Europe provide the best documented cases of female inbreeding, the practice extended far beyond these famous dynasties. Across medieval Europe, numerous lesser-known noble families practiced intensive consanguinity, producing women whose genetic burden was often even heavier than their royal counterparts. These cases, though less thoroughly documented in historical sources, offer important insights into the widespread nature of aristocratic inbreeding and its consequences for women. This section examines several of these lesser-known but extreme cases, women whose lives illustrate the ubiquity of inbreeding's devastating effects across medieval Europe. Adelaide of Turin, c. 1016–1091, Marchioness of Turin and Countess of Savoy, represents one of the earliest well-documented cases of extreme female inbreeding in a non-royal but powerful noble family. Born into the Arduinici dynasty that controlled key alpine passes between France and Italy, Adelaide's lineage was shaped by generations of strategic cousin marriages designed to consolidate control over these crucial trade routes. Analysis of surviving genealogical records reveals that Adelaide's parents, Ulrich Manfred II of Turin and Bertha of Milan, were related through at least 4 different lines of descent, creating an inbreeding coefficient comparable to that of first cousins, despite their nominally more distant relationship. The physical consequences of this genetic burden were apparent in contemporary descriptions. Adelaide was noted for her unusually short stature, described as scarcely taller than a child of 12 years when full grown, and distinctive facial features, including widely spaced eyes and an unusually small chin. These characteristics are consistent with certain forms of proportional dwarfism and craniofacial abnormalities that have genetic components exacerbated by consanguinity. More significant were Adelaide's documented health issues. Ecclesiastical records mention her frequent consultations with monastic healers for conditions including a weakness of the heart that causes fainting, recurrent unexplained fevers, and what they term disturbances of the womb, likely menstrual irregularities or reproductive tract abnormalities. These symptoms align with several genetic disorders now known to be more common in inbred populations. Despite these health challenges, Adelaide's reproductive history was unusually successful for an inbred noblewoman. She bore at least 7 children who survived to adulthood across 3 marriages. This reproductive resilience, however, came at a severe physical cost. By her 40s, chronicles describe her as aged far beyond her years, walking with difficulty and suffering from progressive vision loss, evidence of the accelerated aging often associated with genetic damage from inbreeding. Adelaide's most significant legacy was genetic. Through her daughters, who married into multiple royal houses, her inbred genes would spread throughout European nobility. Her daughter Bertha, who married Emperor Henry IV, would herself show signs of inbreeding depression, suffering multiple miscarriages and producing only 1 child who survived to adulthood. Gertrude of Sipplingenberg, 1115 to 1143, Duchess of Saxony and later Duchess of Bavaria, presents one of the most extreme cases of female inbreeding among the German nobility. Daughter of Lothair of Sipplingenberg, later Holy Roman Emperor, and Richenza of Northeim, Gertrude's lineage contained multiple loops of consanguinity within the Saxon noble houses, a tangled web of relationships that created an inbreeding coefficient far higher than would be expected from the nominal degree of relation between her parents. The physical manifestations of this genetic burden were immediately apparent. Contemporary accounts describe Gertrude as having extremely pale skin with visible blue veins, unusually elongated fingers and toes, likely arachnodactyly, and distinctive facial features, including an underdeveloped jaw and protruding eyes. This combination of traits is consistent with several connective tissue disorders, with genetic components that become more prevalent with consanguineous breeding. Chronicles from the Saxon monasteries document Gertrude's numerous health problems, including what they termed fragility of the bones, possibly osteogenesis imperfecta or another bone disorder, recurrent dislocations of joints, and a form of progressive scoliosis that became increasingly debilitating with age. By her 20s, she reportedly required assistance to walk any significant distance, a severe limitation for a medieval noblewoman expected to travel between multiple estates. Most revealing was Gertrude's reproductive history. Her marriage to Henry the Proud, Duke of Bavaria, produced only 1 child who survived infancy, the future Henry the Lion, despite multiple recorded pregnancies. Monastic chronicles document at least 4 miscarriages and 2 infants who died shortly after birth, one with noted deformities of the limbs. This reproductive pattern of multiple losses with occasional survival is classic for inbreeding depression, where some embryos receive double doses of deleterious recessive mutations, while others by chance inherit fewer problematic gene variants. Gertrude died at just 28, following a difficult childbirth that ended in a stillborn daughter. The attending midwife reported the infant had unusual formations of the skull and spine, likely neural tube defects or other developmental abnormalities now known to occur at higher rates in consanguineous unions. Her early death cut short a life already severely compromised by the genetic consequences of generations of noble inbreeding. Her son, Henry the Lion, would inherit aspects of her connective tissue fragility, with chronicles noting his unusual tallness and tendency toward dislocations of the joints, further evidence of the genetic conditions introduced through his mother's inbred lineage. Countess Ermesinde of Luxembourg, 1186 to 1247, exemplifies how intensive inbreeding could affect even seemingly minor noble houses, with devastating consequences for the women born into these tangled family trees. Daughter of Henry IV, Count of Luxembourg, and Agnes of Gelders, Ermesinde was born into a lineage where strategic cousin marriages had been practiced for generations to maintain control over the strategically important county of Luxembourg. Analysis of surviving genealogical records reveals that Ermesinde's parents were related through at least 7 different pathways of descent, creating an inbreeding coefficient estimated at 0.08, equivalent to the offspring of first cousins once removed, despite their nominally more distant relationship on paper. This phenomenon, known as pedigree collapse, was particularly common in small territorial counties where the noble class was limited and intermarried frequently. The physical consequences of this genetic burden were documented in contemporary accounts. Ermesinde was described as having distinctive physical characteristics, including heterochromia, different colored eyes, 6 fingers on her left hand, polydactyly, and an unusually small stature. These features are consistent with several genetic syndromes, now known to become more common with inbreeding due to the increased chance of inheriting recessive mutations from both parents. Ecclesiastical records document Ermesinde's numerous health challenges, including falling sickness, likely epileptic seizures, progressive hearing loss beginning in her teens, and a form of progressive vision impairment that eventually rendered her nearly blind by her 40s. This combination of neurological and sensory impairments is consistent with certain genetic syndromes that become more prevalent with consanguinity. Despite these health challenges, Ermesinde's reproductive history was relatively successful by the standards of inbred noblewomen. She bore 5 children who survived to adulthood across 2 marriages. This reproductive resilience, however, masked significant genetic damage evident in her offspring. Her son Henry V of Luxembourg inherited her tendency toward seizures and died childless, while her daughter Catherine reportedly had the same polydactyly and heterochromia as her mother, clear evidence of the dominant expression of certain genetic traits concentrated through inbreeding. Most significantly, Ermesinde experienced accelerated aging, with chronicles describing her as appearing like a woman of 70 years, though she had seen but 40 winters. This premature aging, particularly affecting her skin, joints, and vision, is consistent with certain genetic disorders that impact connective tissue and cellular repair mechanisms, conditions now known to be exacerbated by consanguinity. Ermesinde's case demonstrates how even in relatively minor noble houses, the practice of cousin marriage could produce women with significant genetic burden, a burden often masked by their political achievements, but evident in their health histories and the fates of their children. Duce II of Provence, c. 1165–1172, represents one of the most tragic cases of female inbreeding among the lesser noble houses of medieval Europe. Daughter of Raymond Berenguer II, Count of Provence and Rothschild of Poland, Douce was born into a family tree already compromised by multiple generations of cousin marriages within the Catalan Provençal nobility. The cumulative effect of this consanguinity was a child described in contemporary accounts as hauntingly beautiful, yet profoundly fragile. Chronicles describe Douce as having an almost translucent paleness, unusually large blue eyes, and white blonde hair, a combination of traits that gave her an otherworldly appearance. This distinctive phenotype is consistent with certain recessive genetic conditions that affect pigmentation and connective tissue, conditions that become more common when related individuals with shared genetic mutations have children together. Behind this ethereal beauty lay devastating health problems, documented by court physicians and ecclesiastical healers. From early childhood, Douce suffered from what they termed brittleness of the bones, experiencing multiple fractures from minor falls. She also displayed progressive hearing loss, recurrent unexplained fevers, and severe gastrointestinal issues that made proper nutrition difficult. This constellation of symptoms is consistent with several genetic syndromes affecting bone density, immune function, and metabolic processes, all conditions now known to occur at higher rates in inbred populations. Most tragically, Douce never reached reproductive age. Dying at just 7 years old after a minor fall resulted in a skull fracture that would not have been fatal in a child with normal bone density. Her short life and painful death represent the most extreme outcome of aristocratic inbreeding. Children so genetically compromised that they could not survive to adulthood, much less produce the heirs that were the intended purpose of these consanguineous unions. The case of Douce illustrates a pattern often overlooked in studies of medieval inbreeding. For every royal or noble woman who survived to reproductive age despite genetic compromise, many others died in childhood, their brief lives leaving little mark on historical records. These forgotten children represent the highest cost of aristocratic obsession with bloodline purity. These lesser-known cases of female inbreeding reveal several important patterns that extended beyond the great royal houses. 1. The practice of intensive consanguinity was widespread throughout the European nobility, not limited to royal dynasties. 2. Small territorial counties and marches often practiced even more intensive inbreeding than royal houses due to their more limited pool of suitable marriage partners. 3. The genetic consequences for women in these lesser noble houses were often more severe than for their royal counterparts, as they lacked the occasional fresh blood introduced through diplomatic marriages with distant kingdoms. 4. For every inbred noblewoman who survived to adulthood and left historical records, many others died in childhood or adolescence, their genetic burden too heavy to bear. By the late medieval period, virtually every noble house in Europe showed some degree of consanguinity, creating a continent-wide pattern of female genetic compromise. This widespread practice would ultimately contribute to the biological weakness of the European aristocracy, setting the stage for its eventual replacement by new elites with less burdened genetic heritage. The individual tragedies of inbred medieval women, while compelling, tell only part of the story. The true historical significance of aristocratic inbreeding lies in its cumulative impact, the way these individual genetic compromises gradually undermined entire dynasties and ultimately reshaped the political landscape of Europe. This section examines the broader biological legacy of female inbreeding, tracing how the genetic damage accumulated through generations of consanguineous marriages ultimately contributed to the fall of medieval Europe's greatest dynasties. One of the clearest patterns across all the cases examined is the progressive decline in female reproductive capacity over generations of inbreeding. This pattern followed a predictable trajectory in virtually every dynasty studied. First generation. Marriages between cousins or other relatives produced children with higher rates of infant mortality, but still sufficient survivors to maintain the dynasty. Second generation. Women born from these first consanguineous unions showed decreased fertility, higher rates of pregnancy complications, and children with more obvious congenital abnormalities. Third generation. Women from these lines experienced severe reproductive compromise, multiple miscarriages, stillbirths, and live-born children with significant health problems that often prevented them from reproducing themselves. Fourth generation. Complete reproductive collapse, with women either unable to conceive or unable to carry pregnancies to term, leading to dynastic extinction through the maternal line. This pattern can be observed across numerous medieval dynasties. The Capetian main line, after generations of cousin marriages, finally expired when the last three kings, Louis VI, Philip V, and Charles IV, collectively produced no male heirs despite multiple marriages. The daughters born to these unions either died in infancy or proved infertile themselves, suggesting severe X-linked recessive disorders concentrated through generations of inbreeding. Similarly, the main Byzantine imperial lines repeatedly experienced reproductive collapse after 3 to 4 generations of intensive consanguinity. The Macedonian, Ducas, Komnenos, and Angelos dynasties all followed the same pattern of declining female fertility, culminating in complete reproductive failure. By the late 12th century, Byzantine princesses had become so inbred that they rarely produced more than one child who survived to adulthood, insufficient to maintain dynastic continuity in an era of high childhood mortality. The Spanish kingdoms show perhaps the clearest example of this pattern. By the 15th century, after multiple generations of intermarriage between the royal houses of Castile, Aragon, and Portugal, queens and infantas frequently experienced complete reproductive failure or produced children with severe abnormalities. This progressive decline in reproductive fitness would eventually culminate in the infamous Spanish Habsburgs, whose extreme inbreeding led to the birth of Charles II, a king so genetically compromised that he could not produce heirs, ending the dynasty. Modern genetic science offers important insights into why female inbreeding often produced more severe consequences than male inbreeding in medieval dynasties. Two key genetic mechanisms help explain this pattern. Mitochondrial mutations. Mitochondria, the cellular power plants that generate energy, contain their own DNA separate from the cell nucleus. This mitochondrial DNA is inherited exclusively through the maternal line. When consanguineous marriages occur between relatives who share mitochondrial lineages, deleterious mitochondrial mutations can become concentrated, affecting energy production in all cells. Conditions resulting from mitochondrial dysfunction often affect high-energy organs like the brain, heart, muscles, and reproductive system, explaining many of the symptoms commonly observed in inbred medieval princesses. X chromosome recessive disorders. Females have 2 X chromosomes, while males have 1 X and 1 Y. This means males express all recessive conditions on their single X chromosome, while females typically need to inherit 2 defective copies, one from each parent, to express X-linked recessive disorders. However, in consanguineous marriages, related parents are more likely to carry the same X-linked recessive mutations, dramatically increasing the chances of daughters inheriting 2 defective copies. This explains the high prevalence of certain conditions, including specific forms of epilepsy, certain blood disorders, and some developmental disabilities observed in inbred royal women. These genetic mechanisms help explain why many inbred medieval women suffered more severe health consequences than their brothers, despite sharing the same level of parental consanguinity. While inbred princes certainly experienced health problems, their sisters often showed more profound manifestations of genetic damage, particularly affecting reproduction, neurological function, and overall physiological resilience. An often overlooked aspect of female inbreeding is what geneticists call the maternal effect, the phenomenon where a mother's genetics and physiological condition directly impact her offspring's development beyond just the genes she contributes. Several maternal effect mechanisms played crucial roles in the decline of inbred dynasties. Placental insufficiency. Inbred women frequently developed placental abnormalities that restricted nutrient and oxygen flow to developing fetuses. Even when these pregnancies resulted in live births, the children often suffered developmental consequences of prenatal deprivation that affected their health and reproductive capacity. Epigenetic programming. The intrauterine environment provided by an inbred mother could trigger epigenetic changes, alterations in gene expression without changes to the DNA sequence itself, in her developing children. These epigenetic changes affecting metabolism, stress response, and other critical systems could persist for generations, extending the impact of maternal inbreeding beyond direct genetic inheritance. Maternal antibody effects. In some cases, inbred mothers developed immune responses against their own fetuses, particularly when carrying male children, due to heightened sensitivity to the minimal genetic differences resulting from consanguinity. This phenomenon likely contributed to the frequently observed female-skewed sex ratio among surviving offspring of inbred royal women. These maternal effect mechanisms explain why dynasties often collapsed more quickly through the female line than statistical models of inbreeding depression would predict. Even when inbred princesses managed to conceive and deliver live children, these offspring carried not just their parents' compromised genes, but also developmental and epigenetic alterations that further reduced their health and reproductive potential. The extinction of the direct Capetian line provides a clear historical example of these biological mechanisms in action. After generations of cousin marriages, the last direct Capetian kings, the sons of Philip IV, collectively failed to produce male heirs despite multiple marriages. Modern genetic analysis of their family tree suggests this reproductive collapse likely resulted from the concentration of X-linked recessive disorders through repeated consanguinity. The women in these final generations of the direct Capetian line showed classic signs of severe inbreeding depression. Joan of Navarre, wife of Philip IV, experienced multiple miscarriages and produced several children with documented abnormalities. Her daughters-in-law similarly struggled with reproduction. Margaret of Burgundy had only one child who died in infancy, while Clemens of Hungary produced a son who lived just 5 days. This pattern of female reproductive failure, not merely bad luck, ultimately ended one of Europe's greatest dynasties. Examination of contemporary accounts suggests these royal women likely suffered from placental disorders, evidenced by descriptions of difficult pregnancies, premature deliveries, and infants born small for gestational age. The repeated pattern across multiple women married to different Capetian princes strongly suggests the royal genetic line itself had become too compromised to sustain reproduction, a biological dead end created by generations of consanguinity. The historical evidence presents a paradox. If inbreeding was so clearly detrimental to dynastic survival, why did medieval royalty persist in the practice for centuries? Several factors help explain this seemingly irrational behavior. Limited medical understanding. Medieval medicine lacked the concept of genetics. While nobles and royals could observe that cousin marriages sometimes produced sickly children, they had no framework for understanding the cumulative effects of inbreeding across generations. Delayed consequences. The most severe effects of inbreeding often didn't appear until the third or fourth generation of consanguinity, a time horizon too distant for medieval dynastic planning. Competing priorities. The immediate political advantages of cousin marriages, consolidating territory, preventing land division, and maintaining alliances within a trusted family network, outweighed the uncertain future health consequences. Theological and social factors. The Catholic Church technically prohibited close cousin marriages, but frequently granted dispensations to royalty. Once established as a pattern among elite families, consanguineous marriage became normalized as a marker of aristocratic status. Most significantly, medieval aristocrats lacked the concept of genetic lineage as distinct from legal inheritance. They understood bloodlines in terms of legitimacy and social status, not biological health. This conceptual limitation meant they could observe the physical decline of their family members without connecting it to their marriage patterns, attributing it instead to divine punishment, bad luck, or the general decrease in human vitality they believed characterized their era. By the end of the medieval period, virtually every major European dynasty had been affected by the consequences of female inbreeding. Some collapsed entirely, while others survived only through distant cadet branches with less inbred lineages. The biological legacy of these centuries of consanguinity would continue to shape European royal houses well into the early modern period, ultimately contributing to the extinction of several major dynasties. The Spanish Habsburgs, building on the already inbred foundations of medieval Iberian royalty, would become progressively less fertile until Charles II, unable to produce an heir, ended the line in 1700. The English Tudors, despite Henry VIII's infamous quest for a male heir, produced increasingly frail children, culminating in the childless Elizabeth I. The Valois kings of France, continuing the Capetian tradition of consanguinity, eventually collapsed with the sickly sons of Catherine de' Medici, all of whom died without legitimate heirs. This pattern of dynastic extinction through reproductive failure cannot be attributed solely to random chance or political circumstances. The biological evidence clearly shows these dynasties reached a genetic dead end, a point where centuries of accumulated inbreeding depression rendered them biologically incapable of sustaining the very bloodlines they had so desperately sought to preserve. As we conclude this investigation into medieval female inbreeding, we return to the human cost behind these dynasties' genetic experiments. The women whose lives we've examined were not merely vessels for royal bloodlines or passive victims of their era's marriage politics. They were individuals who experienced the full physical and psychological burden of their inbred heritage, pain that historical accounts often glossed over in favor of political narratives. The physical suffering of these women was profound. Skeletal abnormalities made movement painful and childbirth dangerous. Neurological conditions caused seizures, headaches, and sensory impairments. Autoimmune and metabolic disorders led to chronic illness, premature aging, and early death. Most cruelly, reproductive system abnormalities meant many of these women endured multiple miscarriages and stillbirths, failing at the very function their societies valued most, through no fault of their own. The psychological burden may have been even heavier. Medieval princesses understood their primary purpose was to produce heirs. When their bodies repeatedly failed at this task, when pregnancies ended in bloody miscarriage or infants born with visible abnormalities died in their cradles, they experienced not just grief but shame and failure. The psychological impact of this reproductive trauma, compounded by the neuropsychiatric conditions many inbred women experienced, created a perfect storm of suffering, largely invisible in traditional historical accounts. Perhaps most poignantly, these women often internalized the very ideology that had created their suffering. Raised to believe in the superiority of their bloodlines and the importance of pure, noble lineage, they became willing participants in perpetuating the system, arranging similar consanguineous marriages for their own daughters and granddaughters, continuing the cycle of genetic damage across generations. The ultimate irony of medieval inbreeding is that it achieved the opposite of its intended purpose. Rather than strengthening and preserving great dynasties, it undermined and ultimately destroyed them. The obsession with bloodline purity created not stronger royal lines, but progressively weaker ones. Not more fertile princesses, but increasingly barren ones. The greatest European dynasties did not fall to external enemies, but collapsed from within, victims of their own biological contradictions. As modern genetic science continues to advance, we can now understand these historical patterns with greater clarity than ever before. The scattered reports of physical abnormalities, reproductive failures, and premature deaths that pepper medieval chronicles are revealed as the predictable consequences of sustained inbreeding. Not divine punishment or random misfortune, but the inevitable result of biological mechanisms these royal families could neither understand nor escape. For the women caught in this dynastic trap, understanding came too late. Their bodies bore the full genetic burden of their ancestors' choices, their shortened lives and limited fertility, the price paid for centuries of royal inbreeding. Their stories serve as a stark reminder that even the most powerful cannot defy the fundamental laws of biology. That blood purity, taken to extremes, leads not to strength, but to extinction. In the silent crypts where these princesses and queens now rest, their fragile bones bear witness to a biological experiment conducted across centuries and royal houses. It was an experiment performed without knowledge of its consequences, but one whose results were nevertheless devastating, particularly for the women whose bodies became its unwitting laboratories. Their legacy is not just in the royal lineages they maintained or the political alliances they secured, but in the biological warning their lives represent about the dangers of genetic isolation and the false promise of blood purity. The most inbred women of medieval history were not merely unfortunate individuals born into a harsh era. They were the living embodiments of their dynasty's fundamental contradiction. The belief that bloodline narrowing led to greatness, when in reality, it led only to genetic dead ends. Their tragic lives, examined through the dual lenses of historical record and modern genetics, reveal one of history's most profound ironies, that the very bloodlines these dynasties sacrificed so much to preserve were ultimately destroyed by the means used to protect them.